Good morning, everybody. Thanks for coming today. Those online as well. We're excited to have Dr. Sarah Elmendorf today. And just going to do a brief intro. She got her PhD at the University of California, Davis, and has a BA and MA from Wesleyan University. And she is currently a research associate with INSTAAR, or the Institute of Arctic and Alpine Research. And she also has an affiliation with the Department of Ecology and Evolutionary Biology. And before this position, she was a postdoc at the University of British Columbia and a staff scientist at NEON and information manager for Niwot Ridge LGER, which is a really cool, all those places I would love to work at. And her research focuses on climate change impacts on biodiversity and phenology, integrating long-term observations, experiments, and big data. And she's led multiple international ecological synthesis projects and has a particular focus on plants in the tundra ecosystem. So with that, we are excited to introduce Dr. Elmendorf for her talk, Biodiversity and Climate Change Through a Big Data Lens. Thank you.
Thank you guys all for coming today. It is nice to be up here. My office is actually right downstairs, so I feel like I don't come up enough, but welcome down anytime you guys want to journey down to the second floor, similar to people on Zoom. So I just thought I'd start out with a little bit of background. If I can make my slides go. Okay. Okay, here we go. This probably comes as a surprise to nobody in this room or online, but the Earth is warming rapidly. This figure shows the last century or so of change. And on a global scale, 2022 was tied for the 6th warmest year on record. I would say 2023 is not done yet, but it's probably looking worse. It's interesting when I cannot make the click. And that heat is not distributed evenly across the Earth. So this shows the recent temperature trends distributed globally. And what you'll see here is that the polar regions, the tundra in particular, are warming about 2 to 4 times faster than the global average due to a phenomenon called Arctic amplification. And this provides a lot of the motivation for my work. A lot of the talk today is going to focus on these tundra regions. So I just wanted everyone to be familiar with that. I took this figure from the 2018 National Climate Change Assessment because I think it really lays out where the problems are and where the work is to be done right now. So there's a lot of people working on this sort of interaction between carbon fertilization and climate change. But when we start to think about how it's going to affect humans and biodiversity, it gets really messy. So you kind of wind down through this interactions between impacts on biodiversity through terrestrial and aquatic ecosystems, and how they might affect ecosystem services, those changes, as well as human well-being. And these arrows go both directions, admittedly, but it's a quite complicated problem. And I would say I'm one of a large number of people working in this tiny corner of biodiversity, ecosystem services, and human well-being part of climate change impacts. So a little roadmap for today's talk and where we're going. I'm going to talk about biodiversity and climate change, and I'm going to talk about that from a couple of big data perspectives. One is leveraging long-tail data, and I'll talk a little bit more about what that is in a minute. The second part is going to be how we can scale up using big data and thinking a little bit about what we can learn from the fusion of those two approaches, ideally while not like running over some muskox that are hanging out lurking in the road. But first, I want to get on the same page. I really appreciate that Casey, when she invited me, specifically said, don't use a lot of technical jargon and make sure everyone is on the same page before you get into it all. So there's multiple meanings of big data and biodiversity, and I'll just share with you what I mean when I talk about this here. So big data, where do they come from? I love this figure. It's from Christine Laney's, I think, PhD dissertation. I'm not sure if she's ever published in a paper, but it really made me think in creative ways about where those big data come from. So there's kind of 2 places we can think about. We can think about big science, so maybe really big programs like NEON or satellite missions that are a couple of small groups that are just making huge amounts of data, terabytes of data. But there's also a lot of big data that comes out of this long tail science. So we can think about individual researchers or labs. Maybe some of this data is still in file drawers or monitoring programs that are producing vast amounts of data as well, thinking about boots on the ground data. And so there's sort of 2 big sources here, and I'm going to talk about both of those today because I think both of them are really valuable. I sometimes talk about this long tail of science as getting big data the hard way, but they both are really good approaches. So long tail science, what does it look like? For me, a plant ecologist, it often looks like something like this. So this is a graduate student from when I was a postdoc, Tammy, and she was doing this point frame method. So it's like literally standing and looking over a frame and counting how many times a particular pin drops and hits a particular species. It's really great for reproducibility because it's a structured method, but it takes an extremely long time. So that's sort of the top view. You look down, you count these pins, and then it goes the side view. But you can imagine like getting up to a global picture from this is really hard. But this is sometimes where these really big monitoring networks using standardized protocols can come in. So we can think of like a top view and a side view, but then the world view, if we really have different research groups using the same protocol all over tundra regions worldwide, we can start to get more of a global picture of what's going on. The other part I'll tell you about that, it is also a lot of work in addition to collecting the data, and people don't talk as much about this part of it, but I've done a lot of it, so I just wanted to give a shout out to it. So cleaning big data from these long tail programs is really, really time consuming. This comes from a Forbes survey of data scientists, not specific to ecologists, but there was a lot of chatter on ecology blogs around them when this came out, and I think the proportions are about the same. So what it shows here is like what data scientists actually do. So they spend like 60% of their time just cleaning data, maybe another 20% asking people for their data or finding the data, and then a like really small amount of time, 4%, refining algorithms. And sadly, those are almost an exact proportion to what they say they dislike doing. People have this like vision that they're going to go and they're going to, do these like awesome machine learning things, but they spend a lot of time just getting data sets in a way that makes sense to analyze. So I think this is true of ecology as well as ecological synthesis, as well as, economics or whatever else people are doing with big science, data science tasks. And then the second sort of set of definitions I wanted to get on the same page is when you say biodiversity and climate change, what do you mean by biodiversity? And I think this is kind of alienated language, but for whatever reason, ecologists talk about biodiversity always in terms of the Greek letters. So the first thing is alpha diversity, and that is local diversity, or the number of species in a small area. So you can see in this like infographic here, there's like forest species A on this left side of the river, and then another set on maybe the right side of the river, and there are 11 species over, or 11 over here and 14 over here. And so this would have an alpha species richness of 14 and 11 on the right. So we can think about alpha diversity being like species packing in small areas. Importantly, this way of accounting for biodiversity is totally agnostic to the identities of the species in those environments. The second component of biodiversity we often talk about is something called beta diversity, or species turnover. And you can think about that as the difference between two regions, or the number of unique species in areas, depending how you want to do as differences or similarities. So here, for example, like these hummingbirds exist in both places. They don't count towards the beta diversity because it's not different between the two. But over on the left, like we have flamingos, and we've lost flamingos on the right. So that's contributing to the species turnover between the two areas. And we can think about how this might be important in terms of ecosystem services or provisioning as well, because you could have like an example where alpha diversity was the same. You had a corn plantation and then you switched to a pine plantation, and you haven't lost any alpha diversity. There's still one species and one species, but you can think about services provided by those ecosystems might be super different. And then the last bit is gamma diversity, which is, I think, often what the public thinks about when they think about diversity loss. So this is the total number of species either on Earth or in a local area. When we think about global extinctions or local extirpations, that's usually what we're thinking about, like, did the last tiger disappear from an area? Okay, so done with those definitions. We'll start out by launching into some alpha diversity studies and try to understand what's changing with alpha diversity under climate change. And I'll say this form of diversity is actually thought to be quite important to ecosystem function due to a long series of research about biodiversity and ecosystem function, which posits that as you have more species performing more different functions in an area, you might have some sort of increase in ecosystem service due to complementarity. So you can think about like a nitrogen fixer and a phosphorus fixer both living together might capture more resources, or similarly, like an early flowering plant and a late flowering plant might be able to be more productive living together than two that are having exactly the same function. So this diversity ecosystem service often comes from these alpha diversity studies. But we thought, went into the study thinking, well, this framework about the importance of alpha diversity has really been studied a lot, but is it actually changing? It's a question that really people hadn't asked a lot before. And I will start with the punchline, and I will just tell you, alpha diversity or species richness is like remarkably stable over time. So for this study, we went out and we did a literature review, and we just went and pulled out all the plot-based monitoring studies. This is just plants we could find in the literature. And it came out to be 168 published studies in over 16,000 plot-based monitoring areas. And the sum result here was that basically there's no net increase or decrease in plant species richness over time. This shows the effect size in terms of like proportional change, log species richness per decade. And you can see it's exactly centered on 0. So there's some that are increasing, some that are decreasing, but most areas are actually seeing no change in the small plot scale species richness. I didn't find this super surprising. There's not actually a big theoretical reason why we should see increases and decreases in species richness, because that's really about coexistence theory, not so much about what climate change impacts or human impacts would be. But this did cause quite a lot of stir. People were concerned about publishing this, that this would really undermine people's feeling about, I guess, extinction in general. And people were saying, well, if you publish this, people will say there's no biodiversity crisis. And that's not the summary message of this. It's just you really need to think about what the scale is where you're talking about biodiversity loss. Okay. Alpha diversity is only one dimension of biodiversity change, as I mentioned before. So what about species turnover or beta diversity? This slide is to remind me first that I've borrowed liberally from other people's work throughout this talk. So throughout, when I'm using like gray font in the bottom, it's a study that I actually had nothing to do with. It's probably excellent, but I might not be able to answer all the technical questions. Anyhow, a little bit after our study on alpha diversity came out, this great study by Dornelis et al. Came out, and they did a similar thing, taking time series of populations. This is part of the Biotime database, if you're familiar with that, and looking at temporal beta diversity over time. And so this shows Jacquard similarity. So really similar communities are at the top and dissimilar towards the bottom. And you can see, like, it depends a bit on the particular study, but basically everything is getting more dissimilar over time. So as you have these long-term monitoring studies, you see through time a lot of quite a bit of species turnover. The estimates is about maybe 10% species turnover per decade. This was, I should mention, not just a plant-based study, but I think follow-up studies have found roughly the same rates. And the third thing I'll tell you is that species compositional change, or this temporal beta diversity, it's non-random under climate warming. So we're seeing change occur, but in particular directions. And so one particular thing that one might hypothesize happens under climate warming is a phenomenon called thermophilization. And this is the idea that under climate warming, species turnover should lead to a greater abundance of warm versus cold tolerant species. The way people typically study that is something like this. They have a plot, they have 4 species in there, and then they go out and they do some sort of fusion with big data things. So they go figure out where all those species live in the world, and they do a climate niche model. And then they assign some traits to those species. So you could say, like, this particular plot has a lot of these 2 kind of coolish tolerant species. And then they survey it again at time 2 or time and whatever, and they find some sort of species turnover theoretically towards these more warm affiliated communities. And typically that's synthesized down into a single number, which is called this community temperature index, which is the abundance weighted thermal affiliation of the community. So there's been a number of studies that shows this happens. It does not happen everywhere, but we were particularly interested in thinking about thermophilization rates and how those rates might change over broad spatial gradients under ambient climate warming, and then also with experimental warming. We were really interested in looking at this in this unique study system, because we were able to look at these 3 aspects of temperature change where you might be able to understand rates. So the first part of the study was a space for time substitution, where we asked how the thermophily of the resident plants varies among sites arrayed along these really broad spatial gradients in temperature based on latitude and elevation. The second part was taking advantage of long-term monitoring programs and asking whether thermophily tracks historical temperature fluctuations in these long-term monitoring sites. So these sites we haven't manipulated, but the temperature is going up, are the communities tracking that temperature change. And then the last piece was taking advantage of these distributed experiments and asking whether experimental warming induces thermophilization that was similar to that observed over space or time. And to conduct the study, we synthesized data that came from long-term experimental warming and monitoring studies throughout the Arctic and Alpine tundra worldwide. Most of them come from the ITEX network, which is a long-term distributed experiment that I've worked with for a long time. Uses these passive warming chambers that warm the tundra about 1 to 2 degrees, and there are sites distributed worldwide. This actually came from my first NC synthesis that I ever went to. Just to date this child is now like 14 and way taller than me, but it was really my first introduction to synthesis, and it was really powerful and important to me and changed my career. So I hope that ESOL continues to do that for people going forward. So what do we find? So the first thing is this measurement of the thermophilization rates over spatial gradients. So we have over here region, which is more or less latitude, and then elevation within region, sometimes going up or down a mountain. And we found on the left, so this change in community temperature per degree Celsius, so degree Celsius induced by the environment, we found about a little less than half a degree of sort of turnover towards warm affiliated species per degree of warming over these broad spatial gradients. Next, we looked under ambient warming, so sites that were increasing in temperature over time, and we found, again, the same direction. So we see turnover towards more warm affiliated communities, but it's about half the rate that we see over these broad spatial gradients. And this is perhaps unsurprising. These are long-lived species. Species turnover doesn't happen immediately, but it sort of gives us some idea about how quickly we might keep pace with climate change along these, with an expectation that we might eventually reach something like these broad spatial gradients, but we're not going to get there particularly quickly. And then the last piece, we also looked at the degree of warming achieved by these warming chambers and found that the rates with warming chambers were really quite similar to that that occurred under ambient warming over time. So that was sort of good evidence that we're seeing the same rates using both natural warming and experimental warming, and it's about half of that over these broad spatial gradients. The summary message here is that you get much faster rates when you use space for time substitution than if you're examining these same processes using long-term data sets or experiments. So what else changes under experimental warming? It's not just their thermal affiliation of the species. We're going to talk a little bit in this next slide using meta-analysis techniques, again, to look at what's happening in those experimental warming chambers. And this is kind of a confusing slide, but really all you need to pay attention to here is that it's decrease and increase on the left. So things that are up at the top are things that like it warm, and things down below are things that do not like it warm. So the first thing I'll tell you, with experimental warming, we again looked at alpha diversity, this small scale species diversity. It's really stable when things are warmed. So sometimes you get different species there, but you don't get more or less species after you warm an experimental plot. But we did find this compositional change, or temporal beta diversity, was really ubiquitous. So across all these vegetation groups, we saw contrasting increases and decreases. I'll point out a couple of those to you now. So we saw, in particular in green, as vascular plants, and we tended to see increases in a lot of vascular plants, but in particular, these low to tall shrubs, so not like the really teeny shrubs, but ones that are like this to this high, tended to increase when you warmed the plots. We also found over here that we had an increase in dead material in the plot. So litter or standing dead is probably somewhat linked to this increase in vascular productivity. And then last, the losers of climate change in the tundra tend to be the non-vasculars. So mosses and lichens were pretty much declining across the board. It's hard to say cause and effect exactly here. It's just observing what's in the plots, but there's probably likely that these tall shrubs grow up, they dump a bunch of litter, and then it starts to drown out some of the shorter mosses and lichens. Next, we want to look at how vegetation, these big functional groups, were changing with ambient warming. So this is taking all the control plots from those experimental studies as well as monitoring studies tundra-wide. And the first thing we found, this is showing just the change in canopy height. Everything grows taller when you warm it. So you get more and bigger of just about every functional group. The second thing we found, this is just showing directional change, so increasing abundance or decreasing abundance across these vegetation groups. And again, I'll highlight here, these are the shrubs, increased over time in most of the monitoring plots we're looking at throughout the Arctic. The Arctic is mostly warming, so this was perhaps not unsurprising, but again, it's good to have two lines of evidence, both the experiments and the long-term monitoring, showing that we saw these increases in shrubs over time throughout the Arctic. It was really interesting to me. Several years after this particular study came out, I had met, as part of the International Polar Year, a group that was working on trying to investigate some of the same questions by doing structured interviews with the Inuit communities in the north. And there was many questions they could investigate that we couldn't. But one thing that came out convergent is they also had a lot of observations of shrub increases over time by these long interviews with a lot of the elders in the community. Fascinatingly, they also figured out that the caribou taste different now, something I would say we were in no way as the wonky scientists qualified to investigate. But I thought it was a really nice example of sort of convergence. And there were some things that were also different actually between people's perceptions of change and what we document in our long-term studies. So why does this matter, compositional change? In particular, compositional change affects species traits, and traits affect ecosystem services. This is probably true worldwide. I know a little bit more about how this works in the tundra, so I'll just walk you through it here. So when we have vegetation change in the tundra, it has basically two direct effects on ecosystem processes. One is through changing rates of nutrient cycling and carbon storage, which then feed back into climate change. And the second piece is through albedo reflectance. So when you have these tall shrubs poking through the snow, it darkens the surface and actually can change the surface energy balance over tundra regions. Can also insulate the soil at sometimes, so also change permafrost thaw depths. And then again, as I mentioned, there's also some pretty strong effects on subsistence resources. So many northern communities rely on berries for part of their diet. And so having increases in shrubs, particularly berry producing shrubs, can be helpful or not, sort of depending on what the resources are that people are using. So for the next part of this exploration, we started to look at how traits change. So a temporal change in not ecosystem, not just species, but ecosystem function under climate warming. And so we looked at a number of traits that are typically measured in plant individuals or plant species. So for this, we're examining how this warming effects might affect sort of plant height on a gradient from tall to short, leaf dry matter content. So you can think about dry and wet leaves, large versus small leaves, high nitrogen or low nitrogen content leaves, and the sort of thick versus thin leaf spectrum. And the methods we're actually just the same as we use for the community temperature index. We're just swapping out an effect trait for a response trait. So looking at, what attributes of the species might affect how it's changing. Well, what about that species might affect some sort of ecosystem service as we average up under warming. So we're using community weighted mean effect traits instead of community temperature index as our response variable here. So what did we find? The first thing we found, just again, looking at broad scale gradients in summer temperature, is that traits co-vary with these broad spatial gradients in temperature, but the relationship is often depends on soil moisture. These are really complicated plots, but it's just going to show you what the sort of average height is, moisture, size, nitrogen content, and leaf thickness you can think of across this temperature gradient. And I've divided them up here by the wet with the solid dark lines, and then the dry with the lighter lines. And I've shown in light sort of lettering what the main effect is for warm, wet environments. So as you get warmer across these spatial gradients, you get taller plants with wetter leaves. They're larger, they have higher nitrogen and tend to be thinner. But we saw much less responsiveness in these dry communities. We then wanted to look at how much not only we see changes in these traits along these gradients, but how much these traits are changing through time. And so I'll just tell you throughout all of these traits we looked at, the only trait we saw changing consistently over the time in these warming tundra sites was height. So even though we have these big gradients, we're not sort of able to replicate that with ambient warming. We're not seeing the same turnover. This shows the change in community trait over time. So it's sort of the average slope per site. And we looked at it in two ways. One, just taking into account species identity. So that's this community weighted mean. And then the second, also trying to look at intraspecific trait variation, which makes the result a bit stronger here. So we see similar declines within species as across communities. But then every other trait we looked at, it was pretty much centered on zero. We're not seeing any change over time towards these more nitrogen, et cetera, communities. We also looked at sort of the covariation between change in traits over time and how much that site had warmed. And we found that height was the only thing we could see correspondence with. So sites that are warming quickly, we found also had faster increases in height than sites that were not warming or warming more slowly. And then we just went ahead and did like a really big paint by numbers approach to scale this up and ask how much taller is tundra vegetation than it was 40 years ago. So for this, we just kind of married like, this is the sensitivity per degree C. This is how much the temperature has changed in these really broad regions. There was also a moisture piece of it. And then said, well, how much do we think height has changed tundra-wide over basically the last 40 years or so? And it's pretty dramatic. So this is 6 to 12 centimeters estimated increase in height of the tundra community over the last 40 years or so. By this point, we're like kind of feeling tired. So collecting long-tail data is a really uphill battle. You're like hiking up this big hill and it's driving rain and you forget your quadrat and march back down to get it. And kind of for fun, we wrote this blog about how long it takes to collect the data for one of these papers. And the title is, "It Takes a Global Village to Write One Paper." And we estimated it was about 20,000 hours of work to collect the data. And then we just a little math and said that was 1,938 12-hour days of work. I argued that we should do 8-hour days of work for, you know, justice and equity, and people don't work 12 hours a day. But my colleague said it's in the Arctic, there's 12 hours of daylight. So it's only 1,938 hours. So that would take one person 5 years to collect all these data. Fortunately, there was about 1 million co-authors on this paper, so it was only 180 hours of work per person involved in the study. But nonetheless, it's a ton of time. So started thinking about like, we need some more tools in the toolkit. We've been watching plants grow, we've been heating things up, and we need something that acknowledges even with 20,000 hours of work, this global coverage still isn't anything but global. We have these maps and we have like dots, but we're missing really big places. We know all experiments have artifacts, passive warming chambers. They aren't always super effective. Sometimes they're warmer in the beginning of the season or the end of the season. We wrote a review article talking about like poking holes in all of our experimental designs. So we need something that has global coverage, a long-term record, and involves less hiking. So we're like, ta-da, the holy grail. It's remote sensing, obviously. Like this is where we should be going all with our work. And it's great as we sort of age into this part of our research programs where we maybe can't make it back down the mountain as quickly as we could before. But I say it's tongue in cheek, like satellite remote sensing over tundra regions was not invented recently. There are people who have been doing it for a long time to show, for example, like there's a really great relationship between NDVI and biomass, and you can paint these awesome maps of biomass of the Arctic region, places people could never get to. And satellite remote sensing over tundra regions also has shown some really interesting results in terms of change over time. So we're seeing widespread greening evident in the satellite record over the Arctic. So there's something that comes out every year called the Arctic Report Card. The Arctic is literally like graded every year on how much it is greening. So this NDVI signal is increasing over time over much of the Arctic. More recently, it's come out that there's also widespread greening seen in the satellite record over the European Alps. So it might not just be an Arctic tundra phenomenon, but also an alpine tundra phenomenon. I was totally blown away. So like 77% of cells over the alpine tundra are shown to be greening or increasing in NDVI over the Landsat record. And last, we could, go into new wavelengths, not just, the typical NDVI, and think about new developments like hyperspectral sensors. They can potentially measure diversity and traits directly. So as we go out into the shortwave infrared, we can start to say things about water and biochemical traits, in addition to pigments, and sort of thinking not in terms of species, but maybe in terms of optical types, where we can interpret vegetation structure or biochemistry and physiology and phenology, as well, just through looking at hyperspectral images of the landscape. And there's some really exciting developments in this area. I'm not going to go into the methods here, but basically like clustering on these optical types, you can start to look at diversity directly. So you might be able to look at how many different optical types there are in an area and make these maps of alpha diversity. You might be able to look at where the unusual optical types are. And so look at species turnover space, where is the like really high beta diversity in an area. And then sort of pie in the sky, but maybe potentially as we start to get hyperspectral sensors onto satellites, we could monitor trait change or ecosystem impact through change directly. Maybe we'd have these like time series of leaf matter and nitrogen, non-structural carbohydrates through time. But before we get like super ahead of ourselves here, I want to point out that there's some complications here. So satellites indicate more growth over tundra regions. We see that in the GIMS record, but then we look at the MODIS record over the same period and like it's greening too, but there's definitely regions that one shows browning and the other shows greening, or that more show greening and more show browning. So if it was like the perfect method, you would expect to see it exactly the same, regardless of the particular sensor you're using. And so there's sometimes lack of consensus among platforms in the locations and degree of change. And I would summarize that as basically sometimes what you see from space is not exactly reflective of what is occurring on the ground. There's a gajillion reasons for that. And we wrote a review paper outlining some of them. I will tell you, like, don't read that right here, but the really short summary is that remote sensing data from these cloudy and wet and snow-covered locations with extremely short growing seasons can be difficult to interpret. So the satellites are not going by all that often. The growing season is really, really short. But it's also like cheaper and less carbon intensive than traveling there. So there's really pluses and minuses about using a remote sensing approach to study these same phenomena. Which brings me a little bit to the last part of my talk. Closer to home, I'm just going to talk about two projects that are in various stages of completion that are both being run by students, and I think are new exciting directions that we are excited to think about. So the last part of this talk, I will talk a little bit more about the Rocky Mountain tundra, because less has been done here, looking at whether we're seeing that same greening effect over the local alpine. Just to remind you, not only is the Arctic warming, but actually Colorado is warming, the Colorado Alpine is warming. This shows the temperature at the saddle station. So this is up at Niwot Ridge, basically over the Landsat record, with red being the summer temperature and blue being the winter temperature. In both areas, we're seeing significant increases in temperature over time, a bit more in the summer than the winter. And so this is work led by an amazing CU undergrad, soon to be graduate, Miles Moore, and he had just gone through and looked at locations of greening, browning, or no change trends for, we subset it to the Southern Rocky Mountain high altitude tundra over the Landsat record, so about over the last 40 years or so. And what you'll see here is about equal parts of browning and greening. So maybe about equivalent, and then a lot of no change areas as well, despite quite a lot of warming over this area. If you want to know the exact numbers using this particular method, we found 13% increasing and 13% decreasing cells over this area. What's a little disconcerting is we started going down this method rabbit hole, and I'll just show you 8 possible methods here. So this is EVI and NDVI on the left and right, and then just basically 4 different methods where you could try to figure out what the max productivity was from these same vegetation indices. And you get vastly different results. So for example, like the first one I showed you on the left is maybe like half greening, half browning, a lot of no change. And then there's method 4 here, I'll talk about a little bit. We see maybe 1/3 to 40% greening over these areas. And I will tell you, this one is like the one that's most aligned probably with what was done in that study over the European Alps. So I think it's important to think really carefully about not just how we're taking these data from these satellite records, but how we're processing them and how that informs our interpretation of what is happening as we use the Landsat record to detect change. And so the second part of this project, which is not quite done yet, is looking at what are the most robust methods to detect change over time, and how can we take advantage of field data and satellite data to start to look at that. So what Miles is doing now is actually using these 21 years of field-based biomass clippings from the Niwot Ridge LTR, which is just uphill here. They're arrayed in these 88 plots around a grid. They're 40 meters apart, and people have literally been cutting the grass since 1992, when journals apparently couldn't publish in color, up until last year. And they have these small quadrats, and they don't take it right in the plot. They take like an area around it, so you're not mowing the same area. But basically having this long-term biomass record, it spans 20 years of sampling over about 30 years. A few years were missed just to look at which of these satellite basically ways of processing the Landsat record. So each of these is actually in a different Landsat cell. They're small enough, can track what's happening on the ground. And then a second project we're doing along the same lines, taking actually advantage of the same set of long-term monitoring plots, but here we're starting to use NEON data instead of Landsat data. So NEON flies over Niwot. It's a NEON site. It's flown 4 times so far, maybe 5. I haven't seen what they flew this summer. But NEON flies much closer to the ground, obviously, than Landsat does. So it's about 1 meter resolution. And they also have hyperspectral sensors on their plane. So they fly this site, have flown it 4 times so far. So we're trying to leverage these 4 years of plot-based surveys here, not just biomass, but actually what are the plant identities in each of those plots that get monitored each year, and the functional composition of those plots, and sort of marry that with the hyperspectral data to understand, can we use these hyperspectral flights to directly monitor functional change over time? The questions we're looking at is change in functional plant composition. Can we see that from the hyperspectral signal, or you can think of it in terms of functional beta diversity? We want to look, can you see it over space, so over these like 88 plots, or can you also start to see the time series of it and the time series record? We only have 4 years so far, but I will say for hyperspectral data, having 4 years of data is pretty much as big as you would get. It's really rare to have any studies have enough money to refly the same areas using hyperspectral data in multiple years. So I'll just show you a little bit what the spectra look like, because it's kind of visually cool to look at them, and you can toggle back a little bit. So this is 2018. And this is 2019. It's a little subtle, but you can see each of these plots has like a pretty distinctive signature, but they do change a bit between the years, like they go up and down a bit. So we can definitely see some spatial variation, some temporal variation in the spectral signature. So what our student Joe Everest did, and this paper is in revision, hopefully will be resubmitted quite soon. We looked at the dissimilarity in spectral space versus in plant functional space. And if we looked at it for 4 different years, there's a sort of outlier cluster of points here. I'll tell you, the relationship looks pretty much the same with or without them. We decided to keep them in. And these show the correlation and then the significance of a Mantel test. And we see basically this common dissimilarity. So as plots get more dissimilar functionally in terms of their plant composition, the spectral dissimilarity also increases. So we are seeing over space some sort of ability to map the functional similarity or dissimilarity of communities using hyperspectral data. But when we flip it and look over time, so we're going to look at this correlation coefficient here, the relationship is much weaker. So we are not able to track temporal turnover and function nearly so well as we can track spatial turnover and function, which I think has implications for trying to use these for long-term monitoring. And so one of the really tricky things about long-term monitoring when you have one airplane flying to multiple sites is trying to get there at the same time every year, or even when the plants think it's the same time every year. And this is where phenology finally rears its head. So what NEON is trying to do, they're trying to fly at peak season every year, according to the MODIS record, which is shown over here in green. Then sometimes peak season isn't when it's supposed to be. So like the last couple of years, whoops, it's exciting. So sometimes peak season is a bit later than you thought it was going to be. And then when NEON has actually turned out to be able to fly those sites is often way later. So the actual flight window is shown in these oranges and reds, which is kind of getting more towards fall. And then this shows the sort of offset between when they were able to fly and when peak season was. And when we look in the data more, we can actually see the spectral difference between 2017 and any other year is just much higher than the other years. So it's getting really difficult to track these functional changes when it's sort of confounded with a seasonal change that we're seeing when they fly at different times. Nonetheless, I think there's a lot of really cool things to be done with this data, and there's probably like a lot of creativity to go into trying to how to think about how to better align for these temporal offsets in flight times and in order to leverage these hyperspectral data to do what they're supposed to be able to do, which is really track trait change through time. And with that, I'll just give a little summary and then we have some time for questions. Biodiversity is certainly changing with climate change, but bear in mind this compositional change or the beta diversity is the overwhelming element of change. We're not really seeing small scale changes in diversity. Temperature does drive functional turnover for some traits, but not others. Some are staying quite similar, quite stable over time as the climate warms, despite really big spatial gradients in those traits over latitude. And then I would say remote sensing provides huge opportunities to scale up and eliminate these spatial biases we're seeing using site-based monitoring, using long tail studies, but it does come with its own limitations. And so I think we're still working towards the Holy Grail. Some people might disagree. And with that, I'd like to acknowledge, obviously this work came from a ton of people, mostly not me, as well as funding from a large number of long-term monitoring programs and synthesis centers and funding agencies, and happy to take questions. Yeah.
I have two questions. I'll put them both out and let you get through it.
C. Question C.
That is personal area.
Yeah. Yeah.
Can you?
Yeah, I can. So we don't exactly know how fast things disperse. Most of it comes through like either genetics or the paleo record. It does seem like there are some long-term dispersal events because a lot of the Arctic flora is shared along like their species in Niwot that are also in the Arctic. It's weird. It's cool. But I don't think knowing about like the very, very tail of the dispersal kernel necessarily tells you how fast you're going to get new species in. There is a postdoc who is looking at sort of some of the, I don't know if you call it invasion, but borealization of the tundra. It also seems to be happening pretty slowly. So compared to a lot of systems, you don't see a ton of invasive species in the tundra, even up here, like in Niwot, we're pretty close to sources. And I think it's just a very harsh environment. So they're not none, but And tundra may be an exception to that. We're not seeing a ton of invasive species, but yes, invasive species not in the tundra are a super important phenomenon. Most of the turnover, it's a little hard to detect. So whether or not there's a new species that has never been in the system coming in, you have to be very confident that you had a really good survey in year 0, that you're not getting like faux invasions. It seems like most of the turnover that's occurring in our plots is occurring from species that were already in the area thus far. Yeah.
So as ice recedes, is there a seed bank under there?
So some people have germinated stuff from underneath. Yeah. Most of these were not done in primary succession areas. So yeah, but there are some pretty cool old plants that, some that you can actually get frozen tissue from right where ice comes out. I don't know exactly what the germination rates are under areas where the ice is receding, but yeah. Go ahead.
You said that you talk about the 40
Mhm.
That's the method that you're.
It's not exact. So we went on this thing where we're gonna figure out every combination. And it turns out when you think about the whole processing, there's like, how do you align the different sensors? How do you deal with seasonality? There's like, if you really went every single step, it would be like 130 different methods or something. So we haven't gone all of it, but it is that particular method 4 was taking like quantiles of the distribution instead of trying to fit a phenological curve and then adjusting from there. So the issue is kind of when the Landsat sensor goes over, it's this really short growing season, you're probably not going to get a measurement exactly the day of peak. And so how do you figure out from a bunch of measurements that aren't quite at peak what the peak was? So yeah. Yeah. Yeah, I think we do need more studies where we're comparing with ground data. And so we understand there's 2 ways you can do it. Like you can simulate a distribution and then you can delete 99% of your data and figure out what signal you're able to detect, right? If you're missing a lot of time points in it because it's cloudy. So I think that's one good approach. And then I think the second good approach is where we can compare it to a long time series on the ground. That's also valuable. I'll be the first to admit, like long time series on the ground also have their problems. Like we are not mowing down an entire Landsat cell every year. We're cutting like a really small bit of it. So there's some scaling issues there as well. But I think it's good to try all the approaches. And then if they agree, you're in luck because then you have confidence that a particular method both agrees with the ground data and through simulation, you could get basically the same results. Where they don't, then you have more work to do. Yeah.
I feel like I have to ask 2 questions.
You should. 3.
The big science scale. As we're seeing more learning sensitive products online, there's good reason to believe we'll be able to get to tree species identification. Tundra systems
Are harder. You can't get, unless you maybe flew a drone like right over it, you could not get to it. So this is really like, yeah, we are not able to get at alpha diversity, like small scale diversity from tundra from even an airplane. Trees, I think, are more promising. And so maybe this is like application of something where it shouldn't be applied. But the other argument is tundra is really hard to access. So as much as we can figure out how to leverage these remote sensing techniques, I think it's really important to try to do it. Okay.
What I'm excited about. What's next gen?
I mean, I think that the tundra greening thing is still really important, even though we're seeing these trends, to understand where they're occurring, where do they interact with drying trends. So we're seeing, I mean, we went Landsat. There's some reasons to do Landsat because it's smaller scale. And so you have these pixels that are not mixed with like ponds drying or thermocar. So to get unmixed pixels is kind of helpful to understand different processes that might contribute towards increased productivity, or some cases like maybe massively decreased productivity because, I don't know, there's the whole ground slumps and there's no vegetation left. So I do think understanding the productivity change and the vegetation change in the tundra is really an interesting question still. And I think it's, and maybe this doesn't excite other people, but getting down into figuring out what the methods are that allow us to understand what that signal means on the ground, and then to start to think about what processes are contributing to that with a more robust understanding of where change is or isn't occurring. Yeah. Yeah.
Yeah, thank you very much. Regarding your last experiment that you spectral time
Yeah.
and
Yeah.
so I saw like the first correlation.
Yeah.
The best one.
Yeah. So that's the one they flew late, right? It was interesting. Yeah.
So whether this is a external factor or interspecific similarities there that are correlating more with the optimal time.
Yeah. I was thinking about that, and I was thinking actually that the phenology piece might play into that. So if you fly like towards September, there's some things that are a little bit green still, and some things that are senescing more versus more at the peak. I mean, one could see it going either way, because at the peak you're getting flowering plants like tundra. You've hiked in the tundra here. If you've hiked in the tundra here, then you know it's like very showy. So you see a lot of colors. I thought that might be helpful for, it might be correlated to function, but we actually are seeing, yeah, a bit better correlations in the fall. The issue with flying in the fall is people don't want to just use this for like functional dissimilarity. And in theory, if you want to use it to monitor biomass or something like that, that's going to track more with flying at peak time. So ideally you would fly all the time, right? But that is logistically and financially impossible. So I think probably the key is going to be to try to be as consistent as possible, so that, NEON is trying to make this 30-year time series, but that we can track. There's some things you can only measure once a year, and to make sure that that is done at much the same time of year as possible. But yeah, it's interesting. Yeah.
You talked about reading. Do you know what the mechanisms are behind that? Is it extended growing season? Is it more biomass? Yeah.
Yeah. So it is both. Things do tend to get a bit earlier when it warms. Snow is a big piece of the timing as well for the phenology, maybe even a bit more than the warming. I had always thought, like, if there was an earlier phenology, that should lead to more growth because it is such a short growing season. But it does seem like it's a bit decoupled from the phenology, the high biomass years. And I think it's partly that these plants are just extremely temperature limited in their growth. So earlier or not, if it's warm, they will grow more. Yeah. But that is a super interesting question. And I've definitely been trying to disentangle that and some other data sets, trying to look at like, well, does the timing earlier years, do they have like a higher peak? And they don't always. Yeah. Yeah. Might have to do with moisture in some places as well. So sometimes if you start earlier, then you like burn through all the water. So yeah. 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 Good. Okay.
Thank you.
Anyhow,
Yeah.
thanks.